everybody. Hey, welcome to the first session for the 6th International Symposium for ACC&D. These two are kind of pre-sessions, and depending on your background, they're either a primer or a review of two important areas for understanding some of the technology around non-surgical fertility control. We've got Dr. Bob Wheaton, who's going to take us through Reproductive Biology 101. Now, Bob is a seasoned presenter and teacher and deals with veterinary students, so he's had tough audiences, but I know he's a little intimidated knowing that many of you could be giving this talk. So, but I am really pleased to introduce him. He is celebrating his 10th year on ACC&D's board in this summer. And for the last 7 years has been clinical assistant professor at the University of Illinois in charge of their shelter medicine program. So he has probably done more non-surgical fertility control treatments than just about anybody around, counting what's been available to use, the male intertesticular product Zuterin. And so he brings that as well as a master's in public health as background to his position. So come on up, Bob.
Don't applaud, just throw money. All right, how y'all doing tonight? Thank you for being here. I want to thank the Red Acre Foundation for sponsoring this presentation. How many of you have been to one of these things before? Okay, a little less than half, that's pretty cool. It's funny because when I started speaking, I spent a year and a half working for the Global Alliance for Rabies Control, and I really thought that there would be a place for a non-surgical sterilant or contraceptive in the global fight against rabies. And I would speak at veterinary schools, and I would ask students, how many of you have heard of the Alliance for Contraception in Cats and Dogs? And back in 2009, 2010, no hands went up. And then as the years came on, I'd go to a college that I'd never been to before, and I would say, how many of y'all have heard of the ACC&D? And, a few hands would go up. And each year, this is not going to end well. More and more hands would go up, which is really cool. And so I thought, wait a minute, I can't ask this audience how many of you have heard of ACC&D because that's what this is. But just so you guys know about ACC&D, but we catalyze research, we evaluate potential or introduced approaches, and try to advance approaches or products which we believe to have potential to meaningfully extend the reach of population control programs. Our mission is to advance non-surgical fertility control so as to effectively and humanely reduce the number of unwanted cats and dogs in the world. So brief overview of tonight. We're going to talk a little bit about canine reproduction, anatomy and physiology, feline, because there are some differences, and then we're going to look at a couple of ways that reproduction can be controlled. And I'm going to try to leave some time at the end for questions, but like I said, if anybody has a question, just step up to the mic and interrupt me. I think that's probably the best way to do it. So the question I get a lot from veterinarians is why non-surgical sterilization instead of traditional spay and neuter? Well, we know that spaying and neutering requires technical expertise, equipment, time, recovery space, and significant financial investment on the part of either pet owners or government or nonprofit organizations that may subsidize these procedures. These are some pictures from our mobile sterilization unit at Illinois. You can see it on the top left, and I will point out, let me see if I can make that, that works. There is our esteemed board member, Dr. Amy Fisher, helping out. This was a feral cat clinic at Champaign County Humane Society in Champaign-Urbana, and you can see the students doing spays and neuters in the back, and there's up close, and then the prep tables up front. You can see how the animals were all staged. This takes a lot of time and to make it work takes a lot of money. And in many parts of the world, including here in the United States, veterinarians are not available in adequate numbers and or adequately trained to surgically sterilize all the animals that are in need. Some owners and caretakers resist surgical sterilization for social, cultural, or health reasons. When I was working for GARC back in 2010, there was a rabies outbreak in the Dominican Republic, and I went down there and we found a lot of resistance to castrating male dogs in that culture. And so we were successfully able to sterilize a number of them using a Sтерasol, which is the international version of Zudarin, but they did not want the dogs emasculated. So there are cultural reasons as well as health reasons. You've seen, things in the literature about, maybe spaying and neutering at a young age is not so good, and all these studies that have come out. So people are questioning this. There's also a risk of complication. Although high-quality, high-volume spay-neuter surgeries, if you saw the article not too long ago that compared complications in high-quality, high-volume spay-neuter clinics when compared to mainstream veterinary clinics, pretty impressive. The high-quality, high-volume spay-neuter surgeons really know what they're doing. But there are a number of reasons that we seek to expand our options for dog and cat fertility control. And our intent, and as you all probably well know, is not to replace spay-neuter. I was at a conference several years ago. I was actually working with Joyce at the helping out with the ACC&D booth, and a veterinarian came up to me and said, "You're trying to put me out of business." And I said, "No, sir, I'm not." I said, "We're trying to have a tool for your toolbox, but our goal is not to replace spay-neuter. We want to develop these tools that can be used in certain circumstances to achieve the goals of animal population control, extend the reach of those programs, as well as conserve resources." While we focused on advancing permanent non-surgical options, there may be a role in scenarios for pets and or free-roaming animals for contraceptives as well. ACC&D has pursued initiatives in population modeling for free-roaming cats that looks at not just permanent sterilants, but also at contraception with variations on duration and costs that help us look at that as an alternative tool to a permanent non-surgical sterilant. We were initially encouraged by modeling to pursue multi-year contraception for cats. This picture is becoming clearer through additional modeling, some of which will be presented at this conference. And knowing more about the reproductive processes is helpful because some approaches are unlikely to be permanent. I liken them to a slide rule. Who in this room knows what a slide rule is? Oh, I'm surprised. Think about it. Imagine being the pocket calculator, or imagine being the slide rule manufacturer the day the pocket calculator came out. Because the slide rule was great. I used one when I was in high school. I guess I'm telling you a little bit about my age. But when I first started college, that's when the pocket calculator came out. The slide rule is instantly obsolete. And I liken some of these things that we are developing to the slide rule, and eventually the pocket calculator is going to come out. But until it does, some of these things, like a 3-year contraceptive, may be better than nothing. So in order to save more lives, we need more options. In September of 2009, there was a great article in the September issue of Science entitled "A Cure for Euthanasia." And it was mostly about the Michelson Prize and grants, talking about how the quest for a non-surgical sterilant really got a shot in the arm. And I liked a number of things that were said, but this quote out of the article: "A non-surgical sterilant could reduce the global population of homeless dogs and cats, but there hasn't been money to develop one until now," talking about the Michelson Prize and grants. And it's really exciting to see. This was right after I came on the ACC&D board, and it's been really exciting to see the amount of work that's being done in this area, and just really exciting. And this, again, was 9 years ago, but they talked about some of the things that were being looked at then, and you're going to hear about some of them today, as well as during the conference. But even back then, a vaccine that would block the release of sex hormones, you're going to hear a little bit about that. A virus that would genetically silence fertility pathways, a chemical that would destroy eggs, targeted cytotoxins that would destroy cells necessary for the production of sperm and eggs, and a vaccine that could block sperm from entering eggs. This was 9 years ago. And so you'll hear some updates on these as well as some new things in the next couple of days. So the purpose of this brief overview is to give those of you here the background necessary to understand how potential products for contraception and fertility control work. This is an oversimplification of a wonderfully complex system, but the material should serve to orient non-biologists that are here. So we're going to go through each one of these, but the hypothalamus produces GnRH, which has been termed the master reproductive hormone, which stimulates the pituitary gland to produce LH and FSH, luteinizing hormone and follicle stimulating hormone, which stimulate the gonads to produce gametes, sperm in the male and eggs in the female. They also produce hormones, and those hormones released into the bloodstream have a negative feedback on the hypothalamus in order to regulate this. So I could stop there if you want me to. No, I'm kidding. So the brain, the major control center for reproduction is the hypothalamus, and there are specific neurons there that synthesize gonadotropin-releasing hormone, and there are a number of influences that affect that. Light levels, body condition, age, and blood levels of various hormones. Remember I mentioned the negative feedback loop. One of the most interesting things about the brain secretion of GnRH is that it is secreted in pulses and not continuously. And this is really, really important, because if GnRH is not delivered in pulses, it does not have the normal effect on the reproductive system. Interfere with GnRH, and you interrupt all reproduction in both males and females. In fact, not only is reproduction interrupted, but the species-specific reproductive behaviors are disrupted as well. So back to our little diagram here, we're talking about the hypothalamus secreting GnRH, which then stimulates the pituitary, and so on and so forth. So the pituitary, this gland has specific cells, the gonadotrophs, that have receptors for GnRH that bind to the peptide. Once GnRH binds the receptor on a specific pituitary cell, it causes the release of two larger protein hormones called gonadotrophs, luteinizing hormone, known as LH, and follicle stimulating hormone, FSH, which are secreted into the blood. As the brain gives off pulses of GnRH, these pulses reach the pituitary and cause the pituitary to give off pulses of LH and FSH. And again, back to our diagram here, so the pituitary releases LH and FSH, which stimulate the gonads to produce both gametes and hormones. The gonads. When I was in vet school, we had an intramural sports team. We called ourselves the Nads. And our cheer was, "Gonads, gonads." Only the vet students got it. Once the pituitary secretes LH and FSH, they travel in the blood to the gonads, the ovaries in the females and testicles in the male. And these hormones bind to receptors within the gonads. They coordinate the estrous cycle, the heat cycle, of the female and are important in the production of estrogen and progesterone. In the male, FSH and LH are important for sperm maturation and stimulation of the production of testosterone. And when steroid hormones, estrogen, progesterone, and testosterone, are released from the female or male gonads, these hormones travel in the blood, and if you recall in our diagram, they turn off secretion of GnRH, and this is called negative feedback. As you've seen in our little diagram here, the hormone levels affect the hypothalamus and will slow this down. All right. Anybody have any questions at this point in time? Well, we'll carry on. We're going to start talking about the female dog, the bitch. Typically, and just some real general things. Typically, they reach puberty around 6 months of age. They are monestrous, one estrus during the breeding season, and their estrus cycle includes proestrus, estrus, which we commonly refer to as heat, and then metestrus or diestrus, depending on who you read, and then the period of quiescence, anestrus. So unique aspects of dog reproduction include the fact that diestrus, the progestational phase, occurs after every estrus, even if the dog's not impregnated. Bitches will experience a pseudopregnancy lasting the same length of time as an actual pregnancy, unlike most animals which experience a decrease in progesterone as soon as it is determined that the animal is not pregnant. Dogs are monestrous, meaning they only have one cycle per season, which averages 1 to 3 per year, depending on where the dog is located. So the difference here in the non-pregnant dog on this little chart, they develop a period after ovulation if they're not pregnant. Many dogs will develop a false pregnancy. And I've got a little graph of hormones here in a second to show you. But of course, in the pregnant animal, parturition or gestation occurs, or 63 days, and then parturition occurs, at which time they go into a quiet period known as anestrus. The onset of the first estrus in an individual bitch typically is between 6 and 10 months of age. I've seen dogs, when I was in practice, I saw dogs that didn't have their first estrus cycle until 2 years of age. The interestrus interval in the dog is somewhere between 4 and 13 months. Usually about 7 months is average. Typically dogs will cycle twice a year. The anestrus phase of the cycle normally lasts 1 to 6 months. It is marked by ovarian inactivity, uterine involution, and endometrial repair. The anestrus bitch is not attractive nor receptive to male dogs. During proestrus, the bitch becomes attractive to male dogs, but is still not receptive to breeding. She may become more playful, but still will not allow the male to breed her. A blood-tinged vulvar discharge of uterine origin is present, and the vulva is mildly enlarged. Estrogen rises from its basal anestrus levels to peak levels in late proestrus, while progesterone remains at basal levels until rising after the LH surge, which I'm going to show you graphically here in a second. Proestrus lasts from 3 days to 3 weeks, with 9 days being average. When I was in practice, I remember we just used to tell clients, they say, how long is my dog going to be in heat? And we used to say, well, they're a week coming in, a week in, and a week going out, But an estrus lasts, again, 3 days to 3 weeks. But it says, the reference I've got here is average of 9 days. Again, a week coming in, a week in, and a week going out. Estrogens cause an increased turnover rate of vaginal epithelial cells. That's why you see the progressive cornification of cells, that those of you who are veterinarians or have been involved with breeders, you can pinpoint estrus by vaginal cytology. LH, at the end of the follicular phase of the estrus cycle, there's a marked increase in LH over baseline values, and that develops over about 24 to 48 hours, followed by a return to baseline levels. This surge is thought to occur in response to the decline of estrogen levels and increase in progesterone levels. The LH surge triggers ovulation, making it the central endocrinologic event in the reproductive cycle of the bitch. Progesterone levels begin to rise at approximately the time of the LH surge, which is prior to ovulation. Rising progesterone acts synergistically with declining estrogen to reduce edema of the vulva and vagina. During metaestrus, plasma progesterone concentrations are high. They usually plateau 10 to 30 days after ovulation. In the non-pregnant bitch, the progesterone secretion declines slowly and reaches a basal level about 75 days after the start of the luteal phase. And here we see this graphically. So FH goes up and then, or FSH goes up and comes down. Estrogen starts to increase. We get an LH surge, which causes ovulation, and about that time or slightly before, progesterone levels start to increase. So let's talk a little bit about anatomy. The female genital tract includes the vulva, vagina, cervix, uterus, oviducts, and ovaries, as well as mammary glands, which are found on the chest and abdomen. The oviducts are small tubes that connect the ovaries to the uterus. They're labeled here on this little diagram as fallopian tubes. The end of the uterus is called the cervix, which is labeled right there. It separates the uterus from the vagina and provides a barrier against infection. The vagina is a muscular tube that extends from the cervix to the outside and is connected to the vulva, which is the skin surrounding the opening to the vagina. The vulva is the external opening of the female genitalia. An oviduct is connected to each ovary. After ovulation, eggs are transported to the uterus via the oviducts. Secretion of fluid in the oviducts provides a proper environment for survival of the mature egg, fertilization, and the first few critical days of embryonic life. Proper functioning of the uterus and cervix is also required for the establishment and maintenance of pregnancy. In males, the genital tract provides a pathway for sperm and semen. The epididymis connects the testicle to the ductus deferens, which carries sperm to the urethra. Sperm mature and are stored in the epididymis. The accessory sex glands, such as the prostate, labeled there, create the fluid portion of semen. So feline reproduction, a little bit different than canine reproduction. The female is known as a queen. Random poll, how many cat people do we have in the room? How many dog people do we have in the room? Oh, okay. Maybe, I always wondered why they call the female cat the queen and the female dog the bitch. I don't know. And then there's Tom, big old jolly Tom. In the cat, the puberty typically occurs somewhere between 4 and 9 months of age. The estrus cycle is seasonal, typically January to September. Although house cats, because the cycle is regulated by day length, can cycle year-round. In the cat, when I was in practice, I don't know that I could ever tell proestrus from estrus in a cat, although pro-estrus, I guess they don't allow males to breed. They're attracted, and they're vocalizing, but they won't actually breed. And then, of course, there's estrus, where they will accept the male. And in the cat's pretty cool because ovulation is induced by the act of mating, and they can be affectionate or aggressive towards their owners during that time. Cats are kind of different than dogs because they'll continually cycle if they're not bred, and until they are bred, and so we've got a really nice graph to show that. But they'll proestrus will last a period of time, and then estrus, and then they'll kind of seemingly come out, and then they'll come back in. And so one reference I found said that puberty in the cat, which is defined as the first estrus, average of 8 to 9 months, although we know that the cats can come in to heat as early as 4 months of age, depending on the time of year and day length. And cats are seasonally polyestrous, cycling for an average of about 6 days every 2 to 3 weeks from January to mid-October. And they're seasonally anestrous from mid-October through December. That's, again, short days. Now, again, cats that live, closer to the equator or that are maintained under artificial lighting conditions can cycle year-round. The interesting thing about cats are induced or reflex ovulators. The external trigger, usually the mating from a male, stimulates the release of GnRH from the hypothalamus, and this stimulates release of LH from the pituitary within a few hours that causes ovulation within a couple of days after that. Tomcats go through puberty, and that's defined in the male cat as the first appearance of sperm in ejaculate, somewhere between 8 and 12 months of age. And we know that the penis of a male cat, as you can see from these pictures here, is encircled with a number of cornified papillae. People have called them barbs or spines, and again, thought to have something to do with stimulating the female to ovulate. But they're androgen dependent, and they will appear about 6 to 7 months of age in the male cat and disappear after castration. So we see cats that are presented to us for neutering that don't have testicles, and it's kind of a good thing for the students to see, because how can you tell if they're cryptorchid or if they've already been neutered? Exteriorize the penis, and if you've got a nice smooth penis like this, there are no testicles. If you have an abdominal cryptorchid, they'll still produce the hormones to cause the spines to be present. Historically, it was thought that the engorged penis of the tom and or the spines stimulated the cervix of the queen to induce ovulation. But recently, a group demonstrated that queens will not emit the characteristic coital vocalization when the cervix is probed, but will when the posterior vagina is stimulated. They also showed that the erect penis of a tomcat is too short and too large in diameter to reach the cervix of the queen, suggesting that stimulation of the posterior vagina is necessary for ovulation induction in the species. Mature tomcats are capable of mating repeatedly over a 4 to 5 day period without a decrease in sperm number. Man, talk about libido. Graphically here, in contrast to dogs, domestic cats are induced ovulators, stimulated to ovulate in response to mating. Though spontaneous ovulation has been observed in cats. They are prolific breeders, able to become pregnant within days after delivering a litter, and have been demonstrated capable of producing 5 litters in 1 year. Additionally, a cat becomes sexually mature as early as 4 to 5 months of age. And we know that they're seasonal breeders, but again, close to the equator, where they have long daylight hours throughout the year, they have been observed breeding year-round. So here's what happens in the cat. The light blue is proestrus, and so you get these repeated proestrus and then estrus phases, and when they're bred, then they ovulate and they get pregnant. And so cats, you ever been around a cat in heat? Man, I don't know why anybody would want one of those, especially in the middle of the night. Wow. So the female genital tract is somewhat similar to the dog, consisting of the vulva, vagina, cervix, uterus, oviducts, and ovaries, as well, like the dog, of mammary glands. Similar to the dog, the oviducts are small tubes that connect the ovaries to the uterus, and the uterus is separated from the vagina by the cervix, as in the dog, and so things are pretty similar anatomically between cats and dogs in the female. Now in the male, things are a little different, and we already showed you the presence of the spines on the male penis, but notice anatomically, if you compare to our first slide, see how the penis is kind of aiming backwards, whereas in the dog, the urethra comes all the way down and the penis is down underneath. But similar to the dog, the genital tract provides a pathway for semen. The epididymis connects the testicle to the ductus deferens, which carries sperm to the urethra. And like the dog, sperm are stored and mature in the epididymis. And so some similarities, some differences. All right, so let's talk a little bit about, here's our little flow chart that you've seen already, and we're going to talk about some areas of reproductive control. And the first thing we're going to talk about is GnRH vaccines. So, and these next couple of slides are courtesy of Dr. Amy Fisher, one of the ACC&D board members. I really like these slides, and so I continue to use them because they really illustrate a good point here. So she's got a cat named Slimmy. Let's say Slimmy gets a rabies vaccine. Her body makes antibodies against the virus. So if Slimmy is exposed to the virus, these circulating antibodies bind it, so the virus does not ultimately reach where it's supposed to go, and Slimmy does not contract clinical rabies. So Slimmy's pretty happy about that. So let's say Slimmy gets a GnRH vaccine. You know, her body makes antibodies against her own GnRH. So when the hypothalamus releases GnRH, the antibodies bind it. They do not reach the receptors in the pituitary. FSH and LH are not released, and Slimmy is contracepted. Again, Slimmy is happy. So there is such a product. Many of you are familiar with Gonacon. You will hear more about Gonacon during the next couple of days. It, just as a brief overview, it was developed by the USDA, National Wildlife Research Council, and it has been approved for use in white-tailed deer and wild horses and burros. There have been some demonstrated contraceptive effect in other species. And I'm sad to know that my friend and colleague Julie Levy is not here today, because I was going to pick on her because I've got this really nice picture of her playing with cats. She did a really nice laboratory study a number of years ago that showed that Gonacon was pretty effective at contracepting female cats for a period on average of about 3 years. And we, ACC&D, did a field trial on wild-bred cats not too long ago that unfortunately did not show the same efficacy. So GnRH agonists bind and cause downregulation of the GnRH receptors in the pituitary. Now remember how I said early on that GnRH, to be effective, is secreted in a pulsatile manner. When you've got an agonist, it goes to that receptor site and it's not pulsatile. It's there constantly. And so that causes downregulation, which means that the receptors no longer are responsive. And you're going to hear about a product during this conference, Suprelorin, and it is an implant that is given for controlled release. It's a GnRH agonist, and it's been approved in Australia, New Zealand, and the European Union to contracept male dogs. In the United States, it's an FDA index product to treat adrenal tumors in ferrets. And there's been some research done with both female dogs and male and female cats. And I remember at our last symposium back in, 2013? How time flies when you're having fun. But in Portland, Dr. Judith Sampson-French did a really nice presentation on the Dogs with No Names project up in the First Nations Reserve using Suprelorin in those dogs. Gene therapy has been used to treat human disease and basically provides a functioning copy of missing or mutant gene. And for pet contraception, gene therapy would block key molecules that are essential for sculpting working eggs and sperm. Like gene therapy for human diseases, the overall goal is a one-time treatment, in contrast to a drug that may have to be given at regular intervals. And the most common way to deliver gene therapy is to use a virus, which is basically a streamlined nucleic acid harpoon that naturally invades cells. A gene therapy viral vector is stripped of its genes that promote disease and given a working copy of a gene that is mutated or missing in a specific disease. Gene therapy adds a gene, it doesn't replace one. Adapting gene therapy to targeting genes necessary for fertility has been termed vectored contraception or molecular contraception. And I think these are some terms that will probably be debated and potentially come out as we go forward. But you're going to hear a lot about gene therapy in the next 2 days. Chemical sterilization, the use of chemical compounds which destroy and or render ineffective essential components of the reproductive tract. There are two that I'm going to briefly mention, and again, you'll hear more about them over the next 2 days. Zinc gluconate neutralized by arginine, goes by the trade name Zudarin, and calcium chloride. And there's their little logo there as well. These guys work by directly targeting the sperm-producing tissue in the testicle and causes sclerosis of the seminiferous tubules, thereby preventing sperm from being developed. They don't eliminate testosterone as castration would, but they reduce circulating testosterone to some extent because sperm is not being produced, so you're getting less FSH and LH being released. So you do see a reduction in testosterone, but it is not eliminated. And again, that's controversial as to how important that is. Some people feel that testosterone is important in behavior. Other people say not so much. Depends on who you read and listen to. There was a really nice article in the New York Times. Again, I'm sad Julie's not here. Those of you who know Julie, be sure and let her know that I was picking on her. There's the second Julie Levy picture. This is a nice article that appeared in the New York Times back in December of 2013 entitled New Strides for Spaying and Neutering, about Zuterin. There was a Wall Street Journal article about calcium chloride in 2014, Too Many Dogs, A Simple Solution. So the products that you're going to hear about during the next couple of days, I've already mentioned Zuterin. These are some pictures from our Dominican Republic campaign back in 2010. This is me measuring a dog's testicular width to calculate the dose. And here, my friend, Dr. Marcel Guevara, and I were instructing the local veterinarian on the proper administration of Zuterin. Notice the bottom right picture, how Zuterin is injected from the cranial pole of the kidney. And injection technique was determined to be really important, and faulty injection technique was one of the key contributors to adverse reactions. So that's why we were instructing the local veterinarian about proper administration. And contrast that with calcium chloride here, where you can see in this picture they're injecting from the caudal pole of the testicle. I personally have no experience with calcium chloride, so I don't know why the difference, and I hope to maybe find out while I'm here. Similar in terms of how it works, different formulation, and one of the things that as an ACC&D board member, I feel compelled that calcium chloride's not been reviewed or approved by any regulatory agency. And as a result, we feel that its use should be considered experimental. However, I know there are a number of people here at this symposium that have used it. They're going to be talking about it, and I'm looking forward to hear more about it, because with Zuterin not being available, there's really not much out there. Lastly, PZP vaccine, and it's a female-only product. You're going to hear about a product at this symposium. Zonistat has been commercialized for horses, but it's my understanding that zonopollucida vaccines have not, at least yet, worked very well in dogs and cats. But what happens is the anti-zonopollucida antibodies interfere with fertilization by binding the zonopollucida glycoprotein receptors that surround the egg. Zonopollucida is a glycoprotein that surrounds the egg, and it blocks binding and subsequent penetration of sperm. So now basically all the stuff I've talked about and more is available in the ACC&D's ebook. We call it the ebook, Contraception and Fertility Control in Dogs and Cats. You can download it for free. Lots of great information in there. There are product profiles and position papers also on the website that ACC&D has on some of these different products, as well as FAQs. So I want to leave a little time for questions. So thank you for attending tonight. And this is another Amy Fisher slide. Those of you that know Amy know that she loves cats. I don't know where she is. She probably left when I mentioned her name the first time. But I love picking on her. These are two cute little kitties. And again, thank you to the Red Acre Foundation for sponsoring this presentation. So I will now take questions. You're awfully nice. Yes, would you step up to the microphone, please? Not because I'm hard of hearing.
I was curious to know, with some of the chemical and molecular options, what kind of chemical immobilization is required to do chemical castration on dogs using either of the products that you mentioned?
Zuterin or calcium chloride?
Mm-hmm.
Okay, what was the first part of the question?
Do you have to do any sort of chemical immobilization?
Oh,
Yeah.
immobilization. Okay. So I was counting up the other day. I've zooted over 1,000 dogs. And I think I went back and looked. About 30% of them I was able to do without any chemical restraint whatsoever.
Okay.
These are young dogs with the owner sitting there, feeding them treats on their back, scratching their tummy. And because we use a very tiny needle, and yes, there are pain receptors in the skin of the scrotal tissue, but you're talking about like a 28 gauge needle, very tiny, and you just pop that through the skin. There are no pain fibers histologically in the testicular parenchyma. So once you get through the skin, dog doesn't mind it at all. And so the owner's sitting there feeding it treats, scratching its belly. You do a slow injection, and then you wait in order to allow that pressure to equilibrate. Then you withdraw the needle, do the other side. Whole thing, takes maybe 2 minutes. Now, that said, an older, larger dog, you may run into problems just getting him on his back. So my favorite was a combination of, for those of you that are in the United States, I don't know if they have this in Europe, dexmedetomidine, which is an alpha-2 agonist, mixed with butorphanol. A combination, a little cocktail, because butorphanol's got some analgesic effects as well, as does dexmedetomidine. And you can reverse the dexmedetomidine with atipamezole. So they wake right up. The torb's still on board, doesn't really have much of a sedative effect, but does have some analgesic effect. So, so younger dogs you can do without chemical restraint. Older dogs probably require some degree of chemical restraint. The one thing that you have to be real careful, once you get that needle into the testicular parenchyma, if the dog starts getting fractious, you're going to create inflammation and whatnot, and potentially contribute to a complication. So having them being as relaxed as possible is a great thing. So now the other thing people ask about cats, I don't think I would try to inject a tomcat in the balls without some sort of chemical restraint. Would you? I mean, and there, I'm sure there are people here who have been involved with some of the calcium chloride studies in cats, and I look forward to hearing them or hearing about them. But we know that both Zutarin and calcium chloride, and this is kind of getting off of your question, but it's just sort of a corollary thing, that they don't eliminate testosterone. So I don't know what it would do to that stinky tomcat urine smell. And the other thing is, by not eliminating testosterone, how much, fighting is there going to be from a tomcat? I don't know. I've not used it, but, I can neuter a tomcat in 35 seconds when he's under anesthesia, and you're probably going to have to anesthetize him to inject him. It's going to take just as long to inject. So I never personally, and this is, I tell my students, I'm not telling you what to think. I just want you to think about an issue. In my humble opinion, I don't see a place for an injectable sterilant in male cats. But
I guess that's what I was kind of getting to, because there's a certain amount of risk involved whenever you have to use a chemical immobilization drug of some kind with any animal. And at the beginning of your talk, you were talking about some of the risks associated with surgical.
Yes. Yes.
One of the points of trying to get away from that is to remove
No, that's
some of
true.
those risks.
But even think about a feral cat. Let's say we came up with a magical injection that would sterilize a feral cat. We still have to figure out a way to mark them, and there's going to be some marking stuff, I think, coming on in the next couple of days. But the fact is, a feral cat in a trap, you may need some degree of chemical restraint just to give a decent injection. So it may not be general anesthesia. And that was one of the things when I was training veterinarians on Zutarin that I really liked, was you totally obviate the risk from general anesthesia by just giving a sedative. Something like dexmedetomidine, you can reverse. If you get into trouble, you can reverse it. And, the animal's not completely anesthetized. So.
Okay, thank you.
Did that answer your question?
Oh, absolutely.
Okay, good, thanks. Sometimes I see squirrels. Anybody else? Are you kidding me? Come on, Michelle, pick it apart. No questions?
I'm just interested in calcium chloride. It sounds
Can you step up to the microphone, please? This thing's gonna.
I would just like to know a little bit more about calcium chloride. It seems
I would too.
I can
Yeah, I personally have never used it. I'm looking forward to. I know that that there are going to be people talking about it, and I look forward to meeting the people who are using it and finding out more about it. Absolutely.
Thank you.
Yeah, I don't want to dismiss anything just because I don't know anything about it. One of Whedon's laws is mines are like parachutes; they only function when they're open. You know. So I try to keep an open mind. Did I see a hand up down here?
I was waiting to get a friend.
Ma'am?
I was waiting to get a friend.
Oh. Yeah, I guess you don't have to step up to the mic for that. Were you? Am I your friend? Were you waving at me?
Can be.
Okay, thank you. All right. Anybody else? Wow. Now either I did such a good job that there are no questions, or I totally bored you to tears. So I don't know. But if there are questions, yes, ma'am. Good question. I like questions.
I guess, have they evaluated any more of the like long-term behavioral effects of any of the like chemical castration?
I can't hear you.
Sam, sorry, I have a problem with speaking into mics. Have they evaluated any more long-term results, like behaviorally, of the non-surgical sterilization?
Like Zutrin or calcium chloride?
Yeah.
The question is, if you didn't hear it, has there been any evaluation of long-term behavioral effects? There was one paper that I saw, and it's been a while since I've looked at it, looking at the behavioral effects, but it was in free-roaming dogs, so I'm not sure how great that the results would be necessarily applicable to a group of animals that were under much closer observation. The whole debate thing with regarding to sex hormones. If you look back, some of the original data that was published, 20, 30, 40 years ago, very small samples, they were subject to recall bias because people were called after the animal was neutered. Is he behaving better? And, gosh, I don't remember, how badly he behaved, or there might have been a little bit of wish bias, like, yeah, I had him neutered so he behaved better, so yeah, he's behaving better. James Serpell at the University of Pennsylvania has got some contradictory data that shows that sterilized males and females have higher levels of fear-induced aggression, owner-directed aggression, inner-dog aggression, and touch sensitivity. And I haven't talked to James in a while, but he has kindly allowed me to use some of his data in some of my presentations. And I'm not sure if it has been published. I need to get up with him and talk to him about that. But the whole behavior thing is, it's really, really controversial. And I think it's generally accepted that, yes, castration or reducing male sex hormone will reduce urine marking, roaming, and sexual mounting. But a lot of mounting is not sexual. I've got a Dachshund Chihuahua mix that mounts my Siamese cat. Oh, and he's been neutered, by the way. So, I'm not a behaviorist, so I'm not even going to try to go there. But I would like to see some of those studies, yes. So. All right, we got another person coming to the mic.
With regards to Zutrin, what's the current status?
Little closer.
What's the current status of Zutrin usage in the United States?
You will have to come to tomorrow's presentation. Right, Dr. Moldave?
Thanks for the promotion.
Yeah, there are actually two Zutrin. Shameless self-promotion. There are two Zutrin or injectable presentations. Actually, yours is Zutrin, and you're also looking at Suprelerin. And then I'm going to be involved with some other people looking on Tuesday, more forward-looking with Zuterin. And certainly, are you going to be around on Tuesday? Okay.
But the answer is that it's approved.
Into the mic, please.
Stop me if I start to sing. What, the product is not available now. It is approved by the FDA Center for Veterinary Medicine. The last batch of it expired in March. So it is still approved in the US, and it's just not available.
That's her presentation tomorrow. Yeah. Okay. Anybody else? Yes, ma'am. Well, cool. We did get some questions.
Is there any possibility at all of developing a sterilization method that's deliverable by food?
I'm probably not the person to ask. There was a product out there that was being used and given in food. The problem is, particularly in feral cats, and how do you make sure that cat A doesn't eat enough food for cats B, C, and D and gets the proper dose? And so I'm sure that, look at the cell phone, 20 years ago, look how far that's come. So perhaps I don't know. That's well above my pay grade, but it's a good question, but I'm sorry, I can't answer it for you. Joyce is giving me the cut your head off symbol. That's not what you meant, is it? All right. Thank you. I'll be around if anybody has any questions. For those of you that I don't know, come introduce yourself to me. I have extra drink tickets. Just saying. Thank you very much.